students, good morning. Let us welcome to the lecture 12 of Pharmaceutical Organic Chemistry, that is BP 401T. Myself Dr. Sandeep Kumar Bansal, Ramis Institute of Vocational and Technical Education, Greater Noida, would discuss the unit 2nd of this subject, that is determination of configuration of geometrical isomers. So, as far as this unit is concerned, this unit basically deals with geometrical isomerism, that is various methods of nomenclature of geometrical isomers, cis, trans, Ez, syn, anti, and methods of determination of configuration of geometrical isomers. It also deals with conformational isomerism, that is ethane, N-butane and cyclohexane. This unit also deals with stereoisomerism in biphenyl compounds, that is also known as atrope isomerism, and the conditions required for the optical activity. This unit also explains various stereospecific and stereoselective reactions. So, today we will see what methods are available for the determination of configuration of geometrical isomers. So, before proceeding further, let me tell you what we have discussed in our previous lectures. We have discussed what are the isomers, how they are classified, means whether they are having same connectivity or different connectivity, or they are having rotation around the single bond. So, basically there are various stereoisomers, constitutional isomers and the conformational isomers. So, in our previous lectures 9, 10 and 11, we have discussed the conditions required for the geometrical isomerism and the methods of nomenclature that can be implemented to the geometrical isomerism, that was cis trans, syn anti and Ez system of nomenclature. So, today our discussion will be based on the methods of determination of configuration of geometrical isomers. So, further methods of determination of geometrical isomers of configuration. So, we should know, in our previous lectures we have gone that what does a configuration means what conditions are required for assigning the configuration of geometrical isomers. So, we are well aware how the configuration of geometrical isomers is done. Now, after assigning this geometrical configuration, the concern is how to determine the configuration of geometrical isomers. It means how we assign configuration to geometric isomers. What does it mean again? How do we know? For example, if I am having a set of compounds, say for example malic acid, and I am interested to know that whether it will be cis or trans. So, it will be how it can be done. So, there must be some methods, to identify, to determine whether the given set of compounds which configuration it is following. So, my dear students, as such if we see, there are no hard and fast methods for assigning this configuration, but question is still pending. If there is no hard and fast method, Then how to determine this configuration? So, students, we generally use the differences in the physical and chemical properties. What we are using? We are using physical and chemical properties of the compounds to assign this configuration. So, this is the key point that you have keep in mind, that we are using differences in the physical and chemical properties of the molecules to ensure, to confirm that this compound bears that configuration, whether it is cis, it is trans, it is syn, it is anti, it is E or Z. How we can ensure? So, we can ensure once we have assigning the configuration, we have to confirm it, and we are confirming it on the basis of physical and chemical properties. Ideally, we are not looking for properties, we are looking in the differences in the physical and chemical properties. So, this is the, again I am focusing on this line again and again. So, we are following the differences in the physical and chemical properties to assign the configuration. So, in nutshell, we can say that the methods that are available or that can be utilized for the determination of configuration of geometrical isomers are physical and chemical methods. So, we can say physical methods class A and chemical methods as a class B. Now, which physical methods we can use, for assigning the configuration geometric, better to say geometric configuration. So, these are dipole moment, melting point, solubility, stability, action of heat and spectral properties. So, dear students, these 6 physical methods, namely dipole moment, melting point, solubility, stability, action of heat and spectral properties are used to assign the geometrical configuration. Simultaneously, in chemical methods, we can use absolute methods and we can use chemical correlation methods. Now, what is the basic difference between these two methods? In chemical methods, we are asking absolute methods and chemical correlation method. So, in absolute methods, we are subjecting the given compound to various chemical reactions. So, we are destroying the nature of the compound, and this is better to say it is a destructive method. So, we are using destructive methods in absolute method to identifying the configuration. In chemical correlation, we are not utilizing absolute configuration methods, we are not using destroying methods, we are comparing it, we are not disturbing, because the geometrical isomers or any isomers are very precious for the chemistry point of view. So, they are very expensive molecules. So, most of the time we are not in the position to destroy them. So, that is why we avoid the uses of the destructive methods for the purpose of determination of configuration. So, my dear students, you must have listened, you must have understand that what methods we are using for the determination of geometrical configuration. They are physical and chemical methods, and physical methods are having the properties like dipole moments, melting point, solubility, stability, action of heat, spectral properties, and chemical methods are classified as absolute method and chemical correlation method. So, let us see all of them one by one. So, physical methods, first method is the dipole moment. So, this word is very common, and I hope you must have listened this word number of time from your class 10th. So, as all of us know, the dipole moment is a vector quantity. So, this vector word we start to listen from class 8 itself, from class 9, that what are the vector and what are the scalar. So, if we talk about the properties, physical properties in terms of physics, so there are two types of properties known as vector and scalar. So, vector properties possess both the magnitude and direction, but it is about the vector properties, but scalar properties possess only the magnitude. So, if I pick up an example from physics, so we can say that there is example of speed and velocity. So, speed is a scalar quantity, velocity is the magnitude, this vector quantity, because it possesses both the direction and the magnitude. So, similarly dipole moment is again a vector quantity. So, it is having both the magnitude and the direction. Now, how it is going to impact the geometrical isomerism? So, if it is equal in magnitude, remember this is the necessary condition, the dipole moment must be equal in magnitude. If it is equal in magnitude and opposite in direction, remember equal in magnitude and opposite direction, so it will cancel out each other, and therefore, the trans isomers will have the zero dipole moment. Theoretically or ideally, they will have the zero dipole moment. On the other hand, cis isomer possess higher dipole moment, that is value higher than 0. It will depend from compound to compound, it will vary compound to compound, but it is a general statement that cis isomers possess higher dipole moment than trans. Why? If we see this bond is having direction, it is having direction. So, both directions are opposite, and since both the bonds contain CCl, so bond is same. So, therefore, magnitude is same, but direction are opposite. So, therefore, it counts for 0. But if we say cis, this is the direction, this is direction. So, both the molecule will say both the direction will be a net resultant value. So, say that is suppose it is at the center. So, it will be value some value greater than 0. So, therefore, this dipole moment says cis isomers will possess the higher value of dipole moment as compared to the trans isomers. Let us explore this with few more examples. See, if we see cis 1,2 dibromoethene and trans 1,2 dibromoethene, see here the direction is opposite. So, value is 0, direction are same, net resultant value is 1.35. Similarly, if we look upon one more example, trans 1,2 diiodoethene and cis 1,2 diiodoethene. In this case, again the direction are opposite. So, therefore, value of dipole moment is 0, but in this case the value of dipole moment is around 0.75. So, from these example it is very much clear that the cis isomers will have the value of dipole moment higher than the trans isomers. Next comes physical property that we can utilize for the configuration of geometrical isomer, that is melting point. So, as a general statement, cis isomers have lesser melting point than trans. Now, how it is happening? See, this is one example of cis isomer. If we see the shape of the configuration, it is arising like U, but so it is looking like a English letter X. Now, if we try to pack U shape structures they will be packed like this. But if you pack X shape structures, so they can be packed more closely as compared to U shape structures. So, this packing results in the trans isomers in a more symmetrical way. So, packing is more symmetrical in case of trans isomers, but it is less symmetrical in case of cis, and due to the difference in this packing, the cis isomers are having less melting point as compared to trans. Now, look upon one example, malic acid and fumaric acid. Malic acid is a cis isomer, fumaric acid is a trans isomer. So, it is following the shape of X letter and it is following the shape of U shape letter. So, if we see malic acid is having melting point 131, on the other hand trans isomer of malic acid, that is fumaric acid, possesses the melting point 300 degree Celsius. So, this example satisfies the statement that cis isomers possess lesser melting point as compared to the trans isomers. And in support there are some more examples, cinnamic acid, cis isomer is having 68 degree melting point, trans isomer is having 130 degree melting point. Similarly, crotonic acid, cis isomer is having 15 degree melting point, trans isomer is having 72, oleic acid 16 degree cis isomer and 51 is for trans isomer. Next property that we can again use for the purpose of the determination of configuration is cis isomer. As a general statement again, cis isomers are more soluble as compared to the trans isomer, and reason again the poor packing, symmetry. To trans isomers, as we have discussed in our previous slide, trans isomers are more symmetric, but cis isomers are less symmetric as compared. We are talking all the points in a comparative account, we are not discussing any absolute values, we are discussing values in a comparative account. Therefore, trans isomers are more symmetric as compared to the cis, and due to this symmetry, the packing, the trans isomers are more closely packed and cis isomers are loosely packed, and due to this packing the solubility results. So, cis isomers are more soluble as compared to trans isomer due to this symmetrical or asymmetrical packing. And solubility generally we represent in gram per 100 ml. So, this is an example that malic acid is having solubility 79 gram per 100 ml and fumaric acid is having solubility 0.7 gram per 100 ml. So, this example is enough to explain that cis isomers are having more solubility or they are more soluble as compared to the trans isomers. Next property that we can use to distinguish the configuration is stability. Now, let us see what is the general statement. General statement is that cis isomers are less stable than trans. So, first remember this general statement, cis isomers are less stable than trans. Now, what features, what criteria, what parameters are resulting this less stability, and if we look over the structures. So, it is very clear from picture that the reason is the steric hindrance. Steric hindrance means crowding, steric hindrance means crowding. So, if the substituents are present at a cis position, so their volume, the volume occupied in the space will interfere with each other, may interfere with each other if they are very near to each other, and the result is crowding, and this crowding results in the steric hindrance. But if we see trans isomers, they are opposite, they are placed opposite to each other. So, there is less steric hindrance. And if we talk understand this example with the hydrogen, methyl, ethyl. So, space, so which will possess more hindrance, that is ethyl, methyl and hydrogen. So, as the size will increase, it will force, it will produce more and more steric hindrance, and this crowding causes the strain on the cis isomer. And this strain, what it is doing? It is increasing the heat of hydrogenation and enthalpy. So, let us understand what this example means. Now, what is heat of hydrogenation? How it is correlated with the steric hindrance? How it is associated with the stability? So, heat of hydrogenation is the amount of energy that is being released upon the reduction of any olefinic bond. Reduction, we are conducting reduction with the hydrogen, and in the presence of any catalyst like platinum, and olefinic bond is converted into its saturated derivative. So, heat of hydrogenation, again energy released during the conversion of one olefinic bond into its resultant saturated derivative. So, if we look upon the energy release during this procedure is for trans 2 butene, cis 2 butene and 1 butene. So, we can compare the energy released for all these 3 isomers, trans 2 butene, cis 2 butene and 1 butene. So, 1 butene evolve greatest amount of heat when hydrogenated. So, it is least stable. On the other hand, 1 butene must have the greatest energy and be least stable, and trans butene have the lowest energy. So, it is the most stable isomers. Now, this has been represented here in the form of reaction. So, 1 butene is releasing an energy of minus 127 kilojoule per mole, 2 butene cis 2 butene is releasing energy 120 kilojoule per mole, and trans 2 butene is releasing energy minus 115 kilojoule per mole. So, therefore, trans 2 butene is more stable, and stability is reflecting in the form of enthalpy or energy. The same representation has been done on a form of a graph, and we can observe the difference in the energy is 7 kilojoule per mole, 5 kilojoule per mole. So, it is a direct evidence that is correlating that the energy is stability decreasing, and stability is correlated with the form of the steric hindrance. Next parameter that we can use is the action of heat. Due to action of heat, the cis isomer can be converted it easily into the cyclic derivative, that is anhydrides. It is due to its arrangement, and on the other hand, trans isomers can't be converted easily cyclized due to their position. This is one more example that on strong heating cis and trans isomers are easily convertible. Similarly, we can utilize spectral properties. At this stage, you are not very much aware about these UV properties or spectral properties. So, just at this point, just we can recall or we can remember that these values are more for cis, or they are this value is more for trans as compared to the cis. So, this is the excitation of the molecule in presence of the high energy. Similarly, chemical methods we can utilize here, they are absolute method. So, in absolute method, first is the conversion of the compound into its derivative. See here, in absolute method, what we are doing? We are converting the compounds into cyclic derivative. Due to why it is happening? It is happening due to the cis position of the functional groups. If we see this cis position of functional group, enforcing the molecule to be converted into cyclic derivative. On the other hand, trans isomers are not easy to convert into cyclic derivative due to the position of the functional groups. So, this is one example that we can see. Here we see this chlorine and OH both are cis to each other. So, therefore, they are easy to convert into cyclic derivative, but on the other hand in syn it is opposite to each other. So, no reaction is taking place, that they are not getting converted into cyclic derivative. Similarly, cis isomers can be synthesized from a small molecule like malic acid can be converted from the benzene or para-benzoquinone, and cis-cinnamic acid can also be obtained from ortho-amino cis-cinnamic acid. And this is the last point, that we can utilize this stereospecificity and stereoselectivity for the assigning of the configuration. So, this point will be discussed in more detail. There is one more topic in this unit that we will have to discuss, the stereospecific and stereoselective reaction. So, selectivity, stereospecificity, see here cis is converted into Z isomer, trans is converted into, it is trans is converted into the E isomer. So, stereospecificity and stereoselectivity can also be utilized to assign the configuration, and there are very well known examples, Wittig reaction and the Beckmann rearrangement, how stereospecific and stereoselectivity can be utilized for the differentiation of the compounds. And chemical correlation, we can, this is non-destructive method here with the help of the convergence, since this gamma trichloroacetic acid, crotonic acid is converted into crotonic acid and fumaric acid. So, with the help of configuration of crotonic acid, we can correlate its structure with the help of this fumaric acid. So, we can say that since crotonic acid is trans orientation, so fumaric acid must be have the trans orientation. So, my dear students, I hope you must have understand how we can utilize physical and chemical methods for the purpose of configuration of the geometrical isomers. So, we will meet in next lecture. Thank you very much.